Sorry, I thought about that word. That's a whale. When we look at things in anatomy, this basic stuff we're going over, we started with looking at directional terms and learning how to talk right on consults and stuff, and describing where things are, or answering test questions of whether something's proximal, distal, medial, lateral, all of that. But planes also help us. Every view that you look at with imaging, especially in a book picture, a lot of times they're going to give you a little inset or a key that tells you what plane that's in. And if you look at imaging, like MRI, CT, and you don't know which kind of cut you have, you're going to be lost. You'll learn that in radiology. So you need to know that. And so these planes, and we're not going to list, we won't really get into oblique. We're going to do the three basic ones. So if it's right down the middle, like you cut somebody in half, that's a sagittal plane, or sometimes called the mid-sagittal plane. Either one's fine. When you talk about a transverse plane, it's like it's like you cut yourself in half, but it's not, because you can do a transverse plane at any level, right? It's from front to back, or anterior to posterior, at that level. And then when you look at a frontal plane, or what's also called a coronal plane, I always picture like you're standing in a door. You know, if the door was sharp and came down and cut you from front to back, you have an anterior portion that falls this way and a posterior portion that falls that way. Those are the planes. And in reference to that, you also, when they give you what plane it is, you need to know what direction they're viewing it from. You're viewing it down, inferior up or superior down, that kind of thing. So an example, well, I'll show you when we get, I'll point out when we get to some pictures, but a lot of times you see this. I don't know why it does that on that screen. You see this box, and this will be down on the side of a picture. And so what it's telling you is, if you look at the first one there, it says this is a transverse plane of the cerebrum, and the view is superior looking down. So that helps you a lot to see what's going on. This would be your frontal plane with your view, so you're looking straight on. So you're looking at the second one there, you're looking at like you cut me right down through the sensory going this way, and you're looking at my backside of my cerebrum. And then the mid-sagittal plane, of course, just right down the middle. So I'll point those out when we see some other pictures along the way coming up soon. The other thing we have to recognize, we talked about fluid compartments, then we also talked about what's in body compartments. So if you say the dorsal compartment, everything that's there in yellow, really you mean the central nervous system. Yeah, we list them and say, well, it's the brain, brainstem, spinal cord, but that's your central nervous system. So that's the main components of that compartment. But the ventral body cavity includes the thoracic cavity and the abdominal pelvic cavity. And we can further divide these, and we will as we get into those. We'll talk about those more as we get into those sections. When you look at the thoracic cavity up here in the chest, it's also divided into the pleural cavities and then the mediastinal cavity. And so you have the lungs, and then you have the mediastinum that contains, well, basically everything else, the heart, the lymph nodes, the esophagus, the aorta, all of that goes through the mediastinum. Right? So we will be studying the mediastinal compartments and what's in them later, but for now it's just the compartment that's there. And this picture does have that inset I was talking about, where you see it's a transverse plane, and you're looking from inferior to superior. Right? And you see how that can be confusing? I'm getting ahead for other classes. That can be real confusing if you're looking at that picture and you don't know if you're looking down like this or looking up like this. Things get reversed, right? And so that becomes important. So always pay attention to that inset. Right. The abdominal pelvic cavity is separated from the thoracic cavity by the diaphragm, main muscle of breathing. So under our costals, our ribs, you have this diaphragm. It's a physical separation. But when you look at the abdominal pelvic cavity, there's no separation between the abdominal cavity and the pelvic cavity. The separation comes by what we list as in those cavities. But in general, if you look at the dotted line there, there's no tissue separation, but everything below this region is considered to be in the pelvic cavity. Right? So your urinary, right, genital organs, that kind of thing, down there in that pelvic cavity. And then everything that's in there is very close together. So you have to have membranes surrounding these organs. We already talked a little bit about visceral and parietal, just when we were talking about superficial and deep. But a membrane separates things or does other things in the bowels. Right? I've started to digress there. If you look at what the definition is, a mucous membrane is a membrane that opens to the exterior. You know, like this weather, when it's kind of boggy like this, like we're living in Washington State, sort of reminds me of this morning. I get kind of, clogged up in the sinuses because of it. That would be your mucous membranes. They open to the exterior. There are other areas of the body, of course, that open, like your mouth and things, that open to the exterior. But a serous membrane is the same type of membrane by structure, but it's inside. It does not open to the exterior. And that's what's going to surround abdominal pelvic walls, thoracic walls, the visceral organs. And that's where we're going to get the visceral and parietal layers. When you look at a visceral layer, that will be covering the viscera, covering the bowels, covering the lungs. And then you're going to have a parietal layer that's covering the wall, chest wall, the abdominal pelvic wall. And they're separated. Right? So if your bowels are in there and your abdominal wall's in there, you have a parietal layer and a visceral layer, and there's a space, very small space, but there's a space between them that's full of serous fluid. And if you think about things sliding, it's going to help them not to stick together, to slide, decrease friction. Now I use the abdominal wall, but the best example really is looking at the heart, because y'all are doing that, which I think of Mr. Ruggiero, he's pretty cool. Man, that was cold. That was a cold reception. No. I'm happy to have him here, because if not, I would have had to teach that those classes again. I didn't want to do that. I could have done that. I didn't want to do that. Did he tell you his history? He came over from iPad. We stole him right before the job freeze stuff. And he applied and got up here, so that's awesome. He's been around a while and was on active duty. We both don't belong to Daytona. We don't belong to IPAP. We belong to the Department of Anatomy. And we teach into IPAP, or you teach into physical therapy, nutrition, the wasteful combat paramedic course. Wait. Anybody go through that course? There was someone. What did you think of it?
I did it in Fayetteville, not here. It was quick.
That's my problem with it. Now we got digressed. But this is good info. They have y'all heard of that? The combat paramedic course? Okay. Let's see if you agree with me. You can disagree with me. When we first were here, everybody's like, what does this matter? I know. Come on, y'all. Give me a break. It's Tuesday. Because I was here and they said, hey, we want y'all to help us create the anatomy portion of the expeditionary combat medic. Y'all heard of that one? That's what it started out as. And it cratered quick, and I still have this gold CD that they had us build the anatomy course. And when we sent it, they said, well, that's too much. And they said, listen, this is where they set me up to. They said, we're trying to make someone, not a medic, but pretty close to like PA level. In how many weeks?
Well, so what they told me there was 20, because if they make it 21, they have to move your family with you. So they kind of make it a short time.
So they told us, you need to cut down, so I had my anatomy slides from here, they said, you need to cut those down, because we're going to teach A&P 1 and 2 in 2 weeks, 8 hours a day. How did y'all do it?
Yeah, no, it was quick. Yeah, we spent like a week on cardiology. It was,
They literally, like my whole course for y'all's semester, they wanted to do like in a week. And I'm like, how's anybody so I whittled it down. I was military 21 years. Sure, I'll do whatever you say. You want it whittled down, I'll whittle it down. Y'all wish I would do that, but I won't do it here. Because you won't understand anything, and I'm not putting the combat paramedics people went down. I'm talking about the program needs to be redone. That's what I don't like about it. And then, what's the TDA for that position? You know, where are they going? Did you get a job as a combat paramedic?
I was pretty much a nurse in an ER. The only combat I saw was like people with dementia or like kids.
Is there, like, if the assignments people were moving you,
ECM was originally established to be an SFAP component.
yeah.
That were out by themselves. Originally.
And now why didn't we just use what we already have? 18 deltas, we got Navy corpsmen.
Because you can't mass produce stuff. That's why.
I agree. I'm 100% agreement with you, but they tried to rebuild the world based on a paramedic model, and I don't like it. That's my opinion. I have nothing against you.
I'm here now, so it's okay.
I'm glad you made it through it. You got there quick. Things like that come up, and then we can agree or disagree about it. When I went to physical therapy school, it was an identifier, and that's why I wonder if you got that on your record, what do they do with you? Because even though I had a physical therapy identifier, what happened when I got to Fort Benning? They tried to pull me straight out of the Army. You're not going to a hospital. You're a medic. You're coming out with us. I left reception and went and signed in at the hospital, like any good person. Before they could snake me out. Everybody knows what I'm talking about. You ever played the game in the middle? Okay, back to the heart. We can talk about that later. I still have the disc of the original expeditionary one. When you, what's that?
I went through.
Oh, you actually went through it. Cool. It's too fast, too much, too quick. Talk about my class being how fast can you memorize something. That class is how fast can you memorize something while you're on 14 Red Bulls. Fast, yeah. Okay. When we look at the heart that Ruggiero is covering, it has that visceral and parietal layer. Now he's going to break down, obviously, the walls of the heart into different parts, but we're just talking in general. And a good example is if you had a balloon and you stuck your fist in it, right? The inside portion here would be the would be the visceral layer, and then your parietal layer would be out here, and the air that exists between there would be the interstitial, a type of interstitial space, but we call it the pericardial space, right, pleural space, where you're going to have serous fluid that keeps those from sticking together. And so when you name these, we see a very busy slide, but these are just those serous membranes named for the areas they're in. So all three of these main bullets look the same when you look under. We just changed the fact that instead of just calling it viscera, now we're going to say, well, we're talking about the lungs, pleura. So we're going to say the visceral pleura that surrounds the lungs. Or we're going to say the parietal pleura that surrounds the inside of the thoracic wall. Right? Between them, we're going to have the pleural cavity. And that follows suit. When you get to the heart, you have the visceral pericardium, the parietal pericardium, and a space between them called the pericardial cavity. And then the peritoneal area has visceral peritoneum, parietal peritoneum, and then you have the peritoneal cavity. So all the same, but just letting you know where you're located, what we're talking about. We always keep bringing back homeostasis, and we always will, because that's a big part of learning anatomy and physiology especially. And so I'll often mention what happens in different areas we talked about with homeostatic imbalance. And here in these areas where we have that space, if that fluid is lacking in that pericardial space, right, it's going to start to rub and have friction. If it's inflamed, if it gets infected, and we're going to name these like pleurisy, which is inflammation of the pleural, those membranes, or peritonitis, inflammation of the peritoneum. Right, so they can have problems. Another thing we have to cover is retroperitoneal organs. This list is not all inclusive of the retroperitoneal organs, but some of the main ones when we look at it. We have a peritoneal cavity, right? Abdominal pelvic cavity, but that's also your peritoneal cavity anteriorly. But posterior to that, we have a separation, and behind that separation is the retroperitoneal, behind the peritoneal. And it's an area, if you study emergency medicine, you look for blood loss and things, right? Hidden blood loss. But if you kind of drew a basic line following around here, you'd see that it's going to go up like this, and it's going to include, I'm not going to be exact, this is really just a list to make note of. Kidneys, adrenal glands, which are associated with them, pancreas, your duodenum, some of the intestines, the small intestine, and then your ascending and descending colons. We have to pull up different views and kind of show you how that works. For now, it's just a list. Right? For now, it's just what is considered a retroperitoneal organ based on this. We'll get a lot more into that during block 5. Remember, this is just the intro. And then we have a lot of other cavities. Oral cavities, nasal cavity, your synovial cavities, which would be within synovial joints. You have middle ear cavities. You have your ossicles that are in your ear. All of these exist, your eyes, your orbital, all things we discussed. So we're not going to do that here. All right. As we start to finish out this intro portion before we get into cells, there are 2 ways to add more lines to the body, imaginary lines. All right, we already went through the sagittal planes, we already went through directional terms, but when you look at the abdominal pelvic cavity, you can divide it up one of 2 ways. This is the first way. You'll see this in some books, abdominal pelvic regions. In this example, it's a little more specific. You take a horizontal line and run it just below the ribs, which are called costals, and so that's your subcostal line. And then you have a transtubercle line, and you may not know it, but when you look at the trochanters out here, or the tubercles out on the femurs, you can draw a line across them also. And then you do 2 mid-clavicular lines coming straight down through the middle of the cloud. And that makes kind of a tic-tac-toe board, and those regions is in name. Right hypochondriac, right lumbar, kind of name for what's in that area. Right, you will see this, not often, because that's not normally what we use. And so I don't need you to learn this chart with all the regions. We're going to spend a lot more time talking about what we call quadrants. But you do, a lot of things that I talk about, I like you to see it once, so when you see it again, like somebody else decides to bring this up in emergency medicine, you're not clueless when you get there. You've seen it. But questions from me will come, what's in the quadrants? Remember when I talked about expectations? This is me stating an expectation, right? I want you to learn quadrants. So here are your abdominal pelvic quadrants. It's a little easier because we don't have fancy names, and we just draw a plus sign. We draw one line down through the umbilicus vertically, and one line through the belly button or umbilicus there horizontally. This screen is such a mess. One of these days I'll fix it. Probably about the time y'all come back here to teach. Alright, so this is most commonly used. So here's what we need to pay attention to is, well, what's in each quadrant? Right? If somebody comes in and they're, I love to use this term here, I don't know why it doesn't bother you, I don't know why y'all enjoy it. And you get shanked in the left lower quadrant, now we know what might be damaged. I say shanked because if you get shot in the right or left lower quadrant, what possibly do you hit? There's a lot of people in here and go, well wait, did the bullet hit some bone and go up? They start thinking that. And shank is going right through, it's not traveling anywhere else.
What if you shift them?
Shiv them?
Verb.
What's that?
Shake or shift.
What's the difference between a shank or shiv? Huh?
Shift cuts. I think a shift is a tool.
Yeah. I thought it was like in prison. You build something, you wrap some wrap around it, you shake them.
Shake is a verb, shake.
Yeah.
Oh,
With a shiv.
oh my goodness, we're gonna have problems. Oh, let's cover that now. I'm not an English major. I would have never gotten very much. I remember middle school. And they asked me to do something that I never did. Now I'm being a little funny here because I did make it this far. But the one thing in my life I never did in English, and a lot of times I just took a 0 on it, is they used to make us diagram sentences. You ever done that? That's the most ridiculous exercise I ever saw in my life when I was a kid. I'm a math person. I love math. Math makes perfect sense. Now I probably should have done that. Because adjectives, verbs, did you study English? Like English? You know, write papers? I get what y'all are saying now. I only said we're going to have problems because now I got to think about the way I say things. No, I'm kidding. Here we go. Gallbladder. When you look at the gallbladder is in the right upper quadrant. We abbreviate that, if you saw back here, right, RUQ. You can just write that as right upper. I won't do that in here, but it's the right upper quadrant. And so you have the gallbladder, and then you have the right lobe of the liver, right, that is found over in this area. When you go to the left upper quadrant, you have the left lobe of the liver. Right, so the liver spans both quadrants, and some organs do. When you look at the duodenum, it is going to be found in the right upper quadrant, here in blue, and then you have the pylorus, which is the last portion of the stomach. It's also in the right upper quadrant. But the main portion of the stomach, the body, the cardia there, is over here in the left upper quadrant. Pancreas similarly crosses these two quadrants. So you would say the head of the pancreas is there in pink, is in the right upper quadrant. The body and the tail of the pancreas is in the left upper quadrant. And then the spleen is in the left upper quadrant. The only way to get these confused is to not visualize which side you're looking at. Right? So it often helps if you're new to these and you're studying them. You notice I'm doing it when I say it. I know some of you are thinking that's a little Barney-ish, but I'm actually touching that quadrant, so I know which side of the body I'm on. So I'm not throwing the head of the pancreas into the right or left upper quadrant wrong, If you say, well, it's right upper quadrant, then I'm often touching that quadrant in my mind or with my hand or during the test to remind me where I need to be writing down. Some people get confused for no reason on that. If you look at the small intestine, that one's easy because it's in all 4 quadrants. And it goes all the way through. The only confusing part for some people about looking at these quadrants is when you get to the colon or the large intestine, because it's in all 4 quadrants, but it's more specified for what parts of it are in there. Okay, so this is going to sound confusing at first, and I'll explain it. If you look at the right lower quadrant, down here we're going to find the appendix. And then we're going to have the superior portion of the ascending colon. Right, so let's define that. This portion going up of the colon, this is the ascending colon going this way. And then it's going to make a turn. Right, and it's going to go across. It's actually called the hepatic flexure, but that's not on it. It's going to go across, and this is the transcending colon. It's going to the other side, and then it will make another turn at the splenic flexure there, and it will go to the descending colon. So what the slides are saying is if you're looking for the superior portion of the ascending colon, that is in the right upper quadrant. If you're looking for the inferior portion of the ascending colon, that's in the right lower quadrant. And the same goes for the other side. When you say descending colon, you're going to say the superior portion of the descending colon is in the left upper quadrant. But the inferior portion of the descending colon is in the left lower quadrant. You know, getting the verbiage, what it's saying there. When you first read that, especially if you're reading ahead, you start to go, what's it saying here? You just got to think of how that's defined out. And then our kidneys, even though they're really retroperitoneal, are often listed as being in the right and left upper quadrants. So that's all of our intro. And that's where we start to get into cells. That's where we start to get into like new stuff. Not really new stuff because we studied cells before. We all studied it in high school, studied it again in college. We draw out cell models. We make big boards that show cellular stuff on it. So a lot of that's going to be familiar. But when we get into the subcellular organelles and what's going on in them, and what's going on in specific cells like muscle versus nervous cells, we're going to get pretty deep into that, and that'll be the new portion. So the first thing we're going to do is learn again what a general cell is. Right? Because not all cells look the same at all. But what are the cellular components that are basically possible? And we're only going to talk about human cells. We're not going to talk about like prokaryotic cells. That's for micro. That's the fun part of micro, right? That, antibiotics, bugs, good stuff. So what are cells? When you think of cells and you look at being a PA, and I reference things back to sometimes a lot, being a PA is we're studying medicine to treat patients. A lot of us come in here thinking, more emergency medicine type stuff, but think about the average day working the internal medicine clinic at a hospital. And you got to kind of shift your gears because you don't need emergency medicine so much right then. Right? You're going to hospital medicine. And so it's a different way of thinking. If you really get down to the cells, I'm getting kind of clinical here. I like to give you examples of why things are important. When you get down to the cells, every disorder or disease that we're looking at starts at the cellular level. That's really where the problem is. And so it pays to understand how these cells work. Whether we realize it or not, all diseases are rooted at the cellular level. So each one of these cells is going to be a little different. And we're going to discuss the generalized cell, then we'll get into the next block of how they're all different. But there are living units. Collectively, if you go back to our beginning diagram, you say, well, you take cells and you make tissues, and then you make organs, and you make organ systems, and you make an organism. So they're the start of that. And each one of them is a little tiny kind of chemical manufacturing plant. Right? Cells can do a lot of different things. They can take in different things that they need. They can put out different things. But here's our generalized cell. This is what we all saw like in high school, right? Where we have, that's the plasma membrane. The plasma membrane is also called the plasmolemma. It's the outer wall. I don't know, I shouldn't use wall because that takes us back to plants, right? It's the outer portion that surrounds this cell. It's the plasmolemma. Inside of that, you're going to have cytoplasm, right? Which is like a gelatin mold, kind of a jelly-like substance. And inside of that, you're going to have all sorts of structures and what's called subcellular organelles that we're going to find inside of the cytoplasm. And then, of course, we're going to have generally, in a generalized cell, one nucleus. Right? And that's what's going to contain all of our genetic information. So here's the plasma membrane. So we're separating what's outside the cell. In this picture, outside would be out here. And then inside the cell would be here, intracellular. And so there is a physical separation between like the interstitial fluid and the intracellular fluid, and that's called the plasma membrane or the plasmolemma. And it is selectively permeable. We're going to spend a lot of time on that. But it's selectively permeable. I mean, every cell takes in what it needs, puts out what it doesn't need. They all do different functions, and it's selectively permeable, so that the cell really, by using mechanisms of building proteins to go into that cell membrane and stuff, can decide what it's going to let into that cell. or what it's going to let out of that cell. So not everything can get through it. We're going to get deep into it, but, well, I don't want to say that yet. It also plays a role in cellular communication. We're going to get into all that. Cytoplasm. People often confuse. I even had an anatomy instructor recently that was talking about it, and was like reading a question I'd written for, may have been for CBC, and they're like, well, cytosol and cytoplasm are the same thing. No, they're not. Cytoplasm is everything inside that cell, right? The gelatin mold in there. Cytosol is the water. And why am I spending time to point that out to y'all now, other than just say it? Because specifics. When we get to throughout PA school, I'm constantly trying to teach y'all to help you get through this program, not just my class. And a lot of y'all don't get specific in the beginning. Here's the key. There used to be somebody here named Dr. Walters that said this a lot. Words matter. So specific. Like you get into areas like in nerves, and you say, well, what nerve directly innervates this muscle? All nerves branch off of some spinal nerve, right? But they're all named differently. So when you say a word like directly, that means the one that's directly innervating that, not the one it branched off of. You see the technicalities there? Same thing with cellular components. You would say, what is the cytosol of the cell? I wouldn't write it that way, but the cytosol is the water, the cytoplasm is the water and the rest of the structures that are inside of there. So cytoplasm includes the subcellular organelles and the water. Makes sense? Get specific. Start thinking that way. It'll help you. Every word matters, right? You can't. That's one thing I hate about computer testing is I'm a good test taker in regards to figuring things out. I told you that I don't like. I can't write on it. Because what are you going to do with directly? You're going to underline those. Hard to do on a computer. Y'all are used to computer testing now, so I don't know how y'all do it. Either way, y'all seem to do well. Okay, nucleus. Nucleus contains our genetic material, or our DNA. Right? That's actually called chromosomes. Chromosomes are what carry our genes, our genetics. Right? So each chromosome is going to be a single molecule of DNA and a bunch of proteins. Okay? So each chromosome is going to contain thousands upon thousands of hereditary units. This is how we're going to pass on our traits and continue the evolution of our species. Right? We're going to pass those traits on. More to come in genetics. Okay, so just to recap how I set this up, right, we started here, and then the three parts of a generalized cell means you're going to have the plasma membrane, you're going to have the cytoplasm and its components, then you're going to have the nucleus. But when we get to here, what we're going to start doing is defining out now, okay, let's talk more about just the membrane. Let's talk more about the cytoplasm. So the membrane itself has a funny statement here. It's a phospholipid bilayer with associated integral and peripheral proteins. I'm going to underline it now, and I'll tell you why later. It's relatively stable, but it still is fluid, and so in quotes there, that's how you define the plasma membrane. Phospholipid bilayer with associated integral and peripheral proteins. Whether you know what those mean yet or not, we will soon. It's also called a fluid mosaic model. I didn't make these up. I used them, but I didn't make them up. And that's describing the arrangement of the molecules that are part of this membrane. And so what you have is a bunch of phospholipids that are basically like icebergs floating in it. So here's the membrane, and then you have all these phospholipids that are kind of floating in there. Right. So it's a lipid bilayer. Lipids are not water soluble. What is the main solvent in the body? Water. right? I'm gonna get ahead a little bit, just to keep your thinking online as I go through this. If we have a cell membrane that's selectively permeable, meaning it only lets in certain things, and then we have a molecule that comes along in the interstitial fluid that is water soluble, which a lot of molecules are. Can water soluble, let's just say in general, can a water soluble molecule go through a lipid bilayer? Not easily. Right? So where we're going with this is, we're going to look at the bilayer, then we're going to look at how we get things through it later. Right? But for now, we just got to keep in mind, these are lipids that are separating the inside from the outside of that cell. So you're going to have three different things that make up the structure of this membrane. Okay, so the membrane itself consists of phospholipids that form a lipid bilayer. That's what we just said. It is a lipid bilayer made up of phospholipids, but there are also glycolipids and cholesterol there, which are types of lipids, right? Basically. So there's your facts, as we say. Phospholipids, cholesterol, glycolipids. They're part of the membrane. Integral and peripheral proteins are part of the membrane. Right? Well, let's talk about like specifics. I'm really trying to drive a lot of stuff home this morning to make sure you understand. If you look at a plasma membrane that surrounds a cell, you would say that the plasma membrane is made up of a phospholipid bilayer, integral and peripheral proteins. Right? I just repeated myself. And what if I was to ask you, what is the phospholipid bilayer made of? Or let's just say this, what is the lipid bilayer of the plasma membrane made up of? Phospholipids, cholesterol, and glycolipids. I've asked that before. And people always go, I'll ask, what is the bilayer portion of the lipid membrane, or sorry, what does the bilayer portion of the plasma membrane consist of? And they will answer, phospholipids, cholesterol, proteins. And then they'll come into my office and they'll argue with me that proteins are part of the lipid membrane. And I'll agree with you, yeah, they are. You see the difference in it? The question didn't ask that. The question asked, what are the components of the lipid bilayer portion? Right? I know I don't have to say these things to you, but I think it helps you to understand my thinking and some of the IPAP thinking around here. Get used to reading into that kind of stuff. Okay, so then the answer is, well, wait, where do the proteins come in? They're part of the membrane. Well, the cell's going to make proteins. That's what cells do, is protein synthesis. We're going to take from inside and put different proteins in that membrane. We can also take them out. Right? So the bilayer's there, then the cell's putting proteins in it. Now, are proteins generally always present? Yeah. As part of the membrane, as a whole. Okay, hopefully I didn't confuse all that. Glycoproteins are also associated with this membrane. We talked about proteins. So you have glycoproteins, glycolipids. All this means, and I haven't really gotten into the proteins yet, but this is a protein. It's actually called an integral protein. And this is a peripheral protein. And after the break, we're going to get into that. But sometimes you have a carbohydrate group that is attached to a protein. If that carbohydrate group, right, if you take a carbohydrate and attach it to the Y here, attach it to this integral protein, that's called a glycoprotein. Sugary protein, right? Glycoprotein. Carbohydrate group and a protein. If you attach it to lipids, like down here, attach it to this, I didn't draw it straight, this lipid down here, now we're going to call it a glycolipid. So you just have a carbohydrate group that's attached to that. And we only see the carbohydrate groups like that on the outside of the cell. Because later we're going to see they form a role of cell identity. The sugary coating for that cell. so here's your phospholipid bilayer again. It's not, remember this is just part of the plasma membrane. And so you're gonna have phospholipids. Well, right. Phospholipids lined up, and they face opposite directions, right? Here's the heads, here's the tails. So you're going to spend a little bit more time on this, and we'll talk about it on the next slide. But what the heads are facing inside of the cell or outside of the cell, and then the tails are facing towards each other within this actually bilayer. That's why it's called a bilayer. You have these phospholipids on each side. Right? You also have cholesterol and glycolipids there. So if we go and look at those individual phospholipids, we would learn that they are associated with helping things move through the membrane. Things that aren't lipid soluble, got to get through that membrane, so they can help that. And if it's lipid soluble, it can go through the membrane fairly easily. But we also have charges on these. So depending on what charge it is, positive, negative, is it polar, not polar, right? Whether it can get through this membrane. We're just not, we're getting the pieces as we build here. But the bilayer has a phospholipid containing head, which is hydrophilic, and fatty acid tails, which are nonpolar, they're hydrophobic. So if we drop back to the slide, the fatty acid tails that are facing in are hydrophobic, meaning they don't like water. And the hydrophilic head here of that phospholipid, it is hydrophilic. It likes water. Well, that's good because this is extracellular fluid out here, and there's intracellular fluid in here, and both of those are water. So that's how it's set up. Yes?
In between the hydrophobic tails, is that like a hypothetical space, or is there something that would be between those?
Hypothetical. I mean, they're stacked on each other. Like, you mean, do the tails have a space between each tails?
Right.
Not really. I mean, you do have glycolipids in there with it, and cholesterol. But you can almost think of it as, I mean, terrible analogy sometimes, marbling on a steak, and just a solid piece of fat, but it's not really that big. Doesn't that sound good right now? Nice steak. My brother-in-law is a beef cattle nutritionist. I didn't know you could get a PhD and stuff like that. Yeah, he makes a lot of money doing that, I guess. He's the guy you want with you when you go to like the butcher and he has you cut a steak. It takes like 30 minutes. But the steak's good. He cooks the best brisket I've ever had in my life. Yes.
Sorry, for those integral proteins, since they can be all the way through, from outside to inside, does that mean they can be both hydrophilic and hydrophobic?
Yeah, but they're not really. They're just placed in, they're proteins, and so they're just placed in that lipid bilayer. There's no real defined positive and negative side of an integral protein. It's not hydrophilic, hydrophobic, it's just placed in there. It's a great question. That's one of those questions that makes me think how you got to that. Other than going, he said be specific earlier. I mean, it's a good question. I'm not putting, good question. We don't really talk about charges and movement of proteins, only the lipids. All right, you're going to see more of what they do here in a second. We'll have to break it. The other part, we didn't mention cholesterol. There's glycolipids there, right? There's all those phospholipids, glycolipids. We'll do cholesterol and then we'll take a quick break. So cholesterol, if you look down here, this is the cholesterol. Trying to circle here. There you go. So cholesterol is found kind of attached between, it's forming a hydrogen bond with the actual hydrophilic head. And it kind of fills those spaces. We could talk about temperature changes and what it does to this membrane, but we're not going to get into that here. But what we are going to say is the take home for cholesterol, and all I need you to know is cholesterol is part of the lipid bilayer. And cholesterol, look at the first bullet, mediates membrane fluidity. Now, for those of you who like to go through this over and over, you'll see in some other classes maybe you'll spend some time talking about what does cholesterol do when it's warmer versus cooler? And I don't wanna get into that, it's just kind of a distractor for us at this point. I just want you to know it's part of the lipid bilayer and it mediates membrane fluidity. If I ever ask, I'm trying to be very clear, if I ever ask you anything about cholesterol in the bilayer, that's what it would be. What does it do? It mediates membrane fluidity. Whether you understand how it does that or not. Let's take a break. Here's a nice shot. Yeah, I have a my first was an 84 grade, you know, when I was a kid, and it was a piece of junk, but back then, now I've had many, but I bought a 99 that was painted, and redone the interior, and the motor blew it, and this doctor left it. And so I went up there and bought him his motor ball for 5,000. He'd already put like 20 grand in it. But I have, I went 80s now. I have 80s. We call them Centerlines back then, but they're American Racing now. So I have American Racing, the Dodge Chrome wheels on it. Some 33s. Yeah. Those tires, I would love to find they're too loud for me. I gotta go to Holly.
Question we had.
I can't. I talk about selling, every time something else goes on. It's cheaper than people paying new cars. And I can do a lot of it myself. They have upper lower control on it. They have torsion bars, which I had to study torsion bars. When I hit my limit, I threw a chair across my driveway. This car got out of nowhere. And 3 months ago, I'm driving down the freaking road and I'm trying to spider injection in it. And all of a sudden everything got out, stopped, wouldn't start again. And I hauled it down. Just like that. We took the spark plugs out of it, it flooded our shop with gas. We were hydrolyzed by the spider, they fell, flooded drains with gas. And now I'm worried is I could carry a slight this time, but every month, not enough air. Driving down the road, and I'm sitting, I go to Excel, and I got nothing. I'm on the left road.
Yeah.
to the first carbon, or the first, right, so this outline, this OH is up, this OH is down.
I don't know if I've told you guys, I just, 2 years ago, I picked up another bottle.
Right, so this one's saying alpha.
It's like a very good day. The whole back window. But I like to read it.
So it's a big, like,
This is the motor.
what do you stop?
Like, when you start talking about me, grocery stuff, I'm hauling it to shop. I'm not.
But that's in the second carbon of the galactic. So you're making this, structure almost alpha lactic.
You're wasting time and then you know what I call cash on. Buying stuff over there. Which I did last night. I went and bought the most common reason for a random misfire. Spark plugs. And so I changed the ignition coil and the spark plugs. Let's just get it off. It's easy to do. It took me 2 hours. In the rain, my garage is open, but it's still blowing rain. We're changing spark plugs.
Yeah, but like,
They're pretty accessible.
And I got them in there and started up and people always say that. Every gas station goes, but
it's an alkaline, flips it up, right? So would that make that an alkaline?
my son doesn't understand that.
Don't even
He's always breaking down. I need more people to also, but always trying to tell him, you don't have the money and time. You gotta be.
Gotta get the reliable Toyota.
So if it was a
I'm sure he's driving like racing a golf.
that would be, would that be this, so the alpha ice.
The comments are great.
Yeah. You see that in RVs.
On the first part.
Cummins diesels in RVs. They use that all the time.
Oh, yeah.
They have that conversation.
Right, so that's your one. That would on like 4 and like.
That's it. So it's like up to you hear yours.
So if we go to college, so you're like, this is the one.
I'm gonna go fast.
So, you know, you're naming this. So,
Did you really like it?
the second.
Like test days, it's like when you buy it.
It's like a bug. Everything goes wrong.
Well, I mean, actually, to be honest,
When I get another one, I'm gonna get a big class.
So, like, not thinking about okay, so black is
Same day.
so like don't
It's like a band. You're not.
what is your list? So the bar itself,
I just bought it.
what is the alpha, whatever?
Short bed earlier, I found a guy that
It has alpha towards it.
short bed, short bed charge was fine. I went down there, he said,
it's not here, so I guess this is the
That's not
beta,
even
well,
fun.
that's the beta, it's alpha, what happens, they will beta galactose, or alpha galactose is beta. This is no big, these down, this is the beta, this is the OH, so here, still on that one, maybe alpha galactose.
So if I got
What is alpha? This alpha,
no, it looks just like Donna. I drove it today.
right, like, so I'm just trying to
The
think.
head gas.
Okay, so no, like, not lactose, like, obviously, disaccharides are lactose. Like, if we look at it,
But I
like,
just drove it in today because I drove it 40 miles.
OH,
First time doing it out. Doesn't work out.
what is manufactured?
I bought it for $800. He's the original owner. It's not a definitive. I got an knack for
So it's just polysaccharides, multi-class, whatever, so first. So it is the light on the last half of the path that's gonna be.
You run into so many of those people. Like I'm telling so many people, hard to actually get it. Then I had to start asking, you got a great title, not salad, no lids, not rebuilt. Actually, I'll say this. You get there and they got a title that they don't even know who's on the previous title and they got to go sell it. And I'm like, dude, and it's salad.
So, like you said, Sell that car, you sell that.
That's so,
That's right, you said.
yeah, I was in college, it's all good.
so what I'm trying to think of here is like trying to get the full out of that. So like
Just the grocery
for
getter.
instance, so alpha
Like, people wipe up your nut box, so like, how we bought it, you have like, so it's a massive, like, hard to bring up on the wall.
It's still going. The, sorry, you got me talking. How many
Go west, or is it? How to get a set.
I had it recently. I shut down some cars. And he's got
I mean, I can literally do, it's like 2 seconds and you just find the right brand name, you like the board, you literally like mine was like, well, that's why you go.
Someday. When I grew up, so I just I couldn't take it to shot. Yeah. All right. usually do. Okay, we're still talking about the membrane, right? Things get lost at the break, right? The plasma membrane, and now we've talked about the bilayer. Now you say, okay, well, what about the proteins? So proteins that are associated with the bilayer are called integral proteins or peripheral proteins, right? They do different things. But as a whole, you would look at these proteins as helping things move things into and out of the cell. Right? So these proteins are going to look different. We're going to see them throughout the rest of our studies. You're going to see them in biochem. You're going to see them in micro. But different cell types exhibit different intracellular organelles, and they have different proteins on their plasma membrane or associated with it. So these proteins play a large role in the function of that cell, right? They have a big, big role. And so we got to look at the differences. If you say an integral protein, that means it generally goes, we can actually just classify it here and say an integral protein goes all the way through the plasma membrane. It is fully integrated, right? It goes from inside out. Most of them are called transmembrane proteins. So really, you would say, well, integral proteins go almost all the way or all the way through the plasma membrane. What I'm saying is, how about we just say integral proteins are pretty much all transmembrane. They go completely through it. Like a sodium-potassium pump is an integral protein. It goes all the way through. Peripheral proteins are going to be on the inside or the outside of the membrane. Right? We have to list that on here, one side of the membrane. So we go back to the picture and we would say, well, all three of these, let's number them. 1, 2, they look different, but all of these are types of integral proteins. And then inside or outside of the cell, like here, this one is called a peripheral protein. And you see different structures. Sometimes you'll have a peripheral protein. Like in this picture, this is a peripheral protein. It just happens to be kind of associated with this integral that's here. Right, so peripheral are on the inside or outside, and integral proteins go through the membrane. That's all we need to remember for now. Right? But we got to look at the functions of these proteins. When we look at these proteins, especially integral proteins, a lot of them are ion channels. We're going to move things into or out of a cell. We can use an ion channel. We can also move them by, some of these proteins are called transporters. And transporters selectively are going to move something like glucose from the outside of the cell inside of the cell. And then you have what's called receptor proteins. Receptor proteins, we're going to look at pictures of these in a second. Receptor proteins are looking for a specific ligand. So that protein has a receptor on it, and a ligand's a molecule. Like a sodium molecule is a ligand. A ligand is a molecule that has the ability to stimulate that receptor. So that receptor on that protein is looking for a specific ligand or molecule to bind with it. Can also be used for cell recognition, we'll see. Enzymes that are associated with some of these proteins catalyze reactions, like in the bowels, breaking down like disaccharide bonds and things of milk. Or not milk, but anyway. And then some are cell identity. So let's look at what we mean. We have ion channels that are going to take a molecule, and just to generalize, they're going to open up and allow things to go into, I got to remember right over here, into or out of the cell. So that would be an ion channel. A carrier, I forgot to mention it, I used to have about 10 slides on these 2 slides here, these 6 different types of proteins. But I boiled it down and I just went with the picture out of the book. So I used to have these all written out into bullets. And I'm mentioning it because if not, you come back and go, oh, that's small data on the side. On this slide and the next one, I want you to know that small data on the right side of those pictures on here, even though it's small. Fair enough? So you don't ignore that. So that's what I'm talking about with all of these, but I'm just saving space. Because when you get down to a carrier protein, right, you see like carrier proteins are also known as transporters. And so they're going to take something like an amino acid, kill them. You take like an amino acid and you're able to bind to that protein, and then it's going to change its shape and carry it across. Right. We'll talk more about what changing the shape means, but it goes through a change. And then receptors. Receptors are out here on this protein, and they're looking for this ligand to come along. And when a ligand binds with that receptor, it's going to cause a change inside the cell. Right? Like antidiuretic hormone binds receptors in the kidneys. Right? When I said I want you to know what's in the outside of that, what I want you to know is the different type. What's the type of protein? Carrier protein is a trans protein. I don't need you to know the examples. Like, I don't need you to know their antidiuretic hormone. Just, I need you to know that it's a carrier or a receptor. Receptor is identifying something. Yep.
Excuse me, but what's the difference between an ion
An ion
carrier and
carrier and which?
other carriers?
Oh, that's a good question. We just haven't gotten there yet. But, so an ion channel can be open. Some have gates, and there are things that can activate different ion channels. And later we're going to see that uses energy. ATP. Right? We're going to get back to that a lot. A carrier protein uses no energy because it actually undergoes a physical conformational change to bring that in here. And so you'll see more when we talk about it. Can I leave it till then? But the big difference is this uses energy and this doesn't. I'll show you that though. We good? Okay. Enzymes. If a protein inside or outside of that cell, these could be inside or out, have enzymes. Out here is the extracellular. So if you look at lactase, right, or splitting disaccharide bonds so that you can absorb stuff, that happens because of enzymes on the outside of this protein. Linker cells are going to play a large role in structural and shape of cells. As we get into the organelles of a cell, we'll see that there are fibers within the cell that actually give a cell its shape and provide its form. And then you have linker proteins that can go from one cell to the next and actually attach to those and link those cells together. Types of cellular connections, right? And then the last one is cell identity markers. So if you look at cell identity markers, those are generally where you find glycoproteins. Remember back to the carbohydrate groups? So you have a bunch of glycoproteins on the outside. You're going to find cell identity markers on the outside. It doesn't help them to be on the inside. Right? And these are called glycoproteins, and they're important in things like histocompatibility when you talk about blood cells, any cell in the body. Actually, let's re-describe that the way I said it. Cell identity markers on the outside of a cell. Every cell in your body can be identified by your body. If it is not identified, guess what's going to happen to that cell? Your immune system is going to attack it. It's going to try to kill it. So these identity markers help to have recognition.
That's like your APO antigens.
Yeah, that's part of it, for sure. He's talking about blood cells and the antigens on them identify that cell. So that's getting more specific than just saying glycoprotein. But antigens that you find in all these different courses you're talking about now, yes. Collectively, we call that the glycocalyx, the sugary coating that surrounds the cell. Right? So if you looked at a glycocalyx on a cell, you would see you have a bunch of glycolipids and glycoproteins, these carbohydrate groups that are out here. Like I said, they're only on the outside, and what we're going to use these for is cell identity, but because it's a sugary coating, it also helps them to not stick together, which sounds funny because we want cells to stay together, right? We just talked about connecting them. Okay, a lot of things, you gotta work yourself back and forth. Yes, it's going to help for cell adhesion. We can link them together. It provides enzymatic action. It protects that cell. So just like he mentioned, if you look at the RBC blood group system, such as ABO, RH, we all know our blood type in here, most military people do. That's due to the glycocalyx of the red blood cells. And so red blood cells have a sugary coating on them that actually wears out. That's why they're broken down in the spleen and recycled. As red blood cells travel throughout the body, eventually they will wear out and we'll break them back down. And I'm getting a little bit ahead, but how does the body know that that red blood cell is worn out and tired? You know, because it's not walking around like me about 3 weeks from now, coming into class. No, it's a joke, I love you. Red blood cells traveling through, and it's got all these cell identity markers. As it starts to wear out, it starts to lose those. And if it loses enough of those, the body, the immune system will say, okay, it's time, we're going to destroy that cell. Reuse it. We're going to break that cell down. Okay. Same thing happens if you give somebody a transfusion that's not the same blood type. I don't want to get ahead, that's fine. Okay, we're going to try to destroy that. A sperm cell, that's unique. A sperm cell can actually identify an ovum. Remember, a sperm cell wants to fertilize the ovum when it gets there. And so a sperm cell, I don't know how it knows what it's looking for, but it finds the ovum by looking at its glycocalyx. It's traveling along through the uterus, through the fallopian tubes. It gets to the ovum there and goes, that's a nice looking glycocalyx. And oh, I was leading up to that. Look who's talking now, right? You ever seen that movie? Anyway, it's pretty amazing. The sperm cell not only can recognize where it needs to be when it gets to, didn't even know where it's going, didn't even have Google. And it's going to make it there a lot of the times, right? We know that by all the accents. And it gets there. Not only that, when it gets there, it goes, oh my goodness, I'm getting into repro. There's a hard wall around this ovum I can't get through. Oh, lucky for me, I threw some enzymes in under the hood. About to sit in the trunk, but when you study repro, it's in the front. So. You gotta drive a, what's that truck now? Ribbon? You gotta drive a ribbon. Have the trunk in the front, right? Okay. Got way off. Ligatures, no. Liver phagocytizes worn out blood cells also, right? That's what that word means. Effet means worn out, right? So we start to lose the glycocalyx. Alright, let's get out of that before I get in trouble. Okay. Movement across membranes. That's what we're talking about. The whole reason we're learning a lot of these proteins and defining what types they are is so that we can see what they do. So when you look at any given cell, it is bathed in extracellular fluid at all times around it. Well, extracellular fluid, or if you want to say interstitial fluid, which is the type of that, is full of molecules. Sodium, potassium, chloride, glucose, amino acids. All these things are found in that interstitial fluid or extracellular fluid. And the cell is going to say, well, I need some of that. I need that into my cell. And so the cell has to take what it needs out of the interstitial and then extract or put out what it doesn't need back into that. The waste products go back into out of the cell into the extracellular fluid so it can be moved away. And we said that's called selectively permeable earlier. Okay, so all cells are selectively permeable for what they want put in them. But we're building on that. We're learning how this happens now, not just saying it. So when you look at substances that are nonpolar, they don't have a big charge, and substances that are lipid soluble, they can move through the plasma membrane fairly easily. They can go right through it, right? A lipid goes into lipid. You drop lipid dish soap right into water, what does it do? It beads up, right? Oil into water doesn't mix. Same concept here. If it's lipid soluble or it's nonpolar, it can generally go through that membrane without needing a carrier or needing an ion channel. It can just diffuse through it. If you think about it logically, when you're studying lists, a lot of people study lists, but I always look for things that can help me remember not only the list, but the anti-list, right? What's the outliers? Because the outliers often get active. Oxygen, carbon monoxide, fats, alcohol, fat-soluble vitamins. Most of those we already would agree before we even walked in here today, we gotta have. We need it in every tissue. Do we need oxygen everywhere? Should it, if the oxygen's needed everywhere, should it be able to freely flow everywhere in the body? Yeah. It's a kind of a special case based on its size and no charge, but carbon dioxide, we need to get rid of waste products. Yeah, if not, we're gonna die. We can't build up waste products. Fats are not a bad thing, right? They are in the news, they are in our lives, but there's a lot of good fats, but we need fats for a lot of things. Neural transmissions, I could name them forever. And then alcohol, depends on who you are. I mean, you may need it to transfuse it easily, transfuse, anything use that word. And then fat-soluble vitamins, and I threw that one up there because they move through into the cell fairly easily. When you look at the vitamins, A, D, E, K, or ADEC, I always said, they are fat-soluble. This is why, if you take a bunch of vitamin A, or there was a kick a few years ago, people were taking 10,000 units of vitamin D a day to help prevent cancers and stuff. I don't know if that works, I just know they were doing it. But any fat-soluble vitamin, because it can cross the plasma membrane easily, it's just going to build up in your cells. That's going to hurt you, especially like large amounts of vitamin A that build in your cells. If it's water-soluble vitamin, it can't get through there as easily, and the best example for that is vitamin C. Right? You can go over to GNC today, buy the largest bottle of vitamin C they have, and they taste good. You can sit here and eat them in class to stay awake in anatomy. You can eat that whole bottle in the next 30 minutes. It's not going to hurt you. Where is it going? You're right. Why? I'm using this as a good example. Did we use some of that vitamin C? Yeah. The cell's selectively permeable, and because it can't get through that wall, it's not a fat-soluble vitamin. The cells will only take in what it needs. The rest of it will be filtered out by the kidneys, and you'll be peeing orange. Won't kill you. Go and eat that same size bottle of vitamin A, or even much less than that. You're going to be at the hospital, right? I'm talking a lot for no reason about it, but it helps to explain fat-soluble vitamins versus water-soluble. Okay. So then how do we do it? If this membrane exists, then how do we get things across? We already know the cell can put proteins in there to get things across, but how? Right? This is where we get into concentration gradients. In Texas, sweet tea. Right? I know other states do too, but I'm only talking about Texas, right? If I go to a restaurant and I order sweet tea, and you bring me sugar and a glass of tea separate, right? I don't even want it. I don't even want it. That tea needed to be warm or hot when you put that sugar in, and there needed to be lots of it. I know. My girls and my whole family disagrees. They all drink unsweet tea. It's the bitterest thing I've ever had in my life. You want some good sweet tea? I like Bill Mill. I don't like Bill Miller's barbecue, but I love their sweet tea. Raising Cane's has some good sweet tea. Yeah. Why did I say sweet tea though? Why don't I can put as much sugar as I want in that iced tea they brought me. That's what they're thinking. Is it the same? No, can I get it to diffuse? Bring it back to anatomy. Can I get it to go into into solution? No, right? Because of concentration gradients, right? We can change. That's talking simple diffusion. That's why I use sweet tea. If you take an ice cold glass of tea and you dump sugar in it, by simple diffusion, some of that's going to diffuse into solution, right? But that's still a concentration gradient. There's more tea than there is sugar, it will diffuse. And you can change that. If you warm it up, you can change how much diffuses. I'm using this example because we've probably studied that in classes before. We understand simple diffusion, right? But simple diffusion is a concentration gradient, more or less. You have more, anyway, I'm going to stop there. Inside, when you look at cells, the plasma membrane, it's hydrophobic, right? So anything that's water soluble, not getting through there without help, right? And that includes, so some molecules can passively diffuse. Anytime you hear passively, that means that it was not assisted, it didn't use energy, it just happened, right? It's able to do that. So if it's lipid soluble, as we saw earlier, if it's small enough to pass through, or it has to be assisted by carrier molecules. Carrier molecules includes a lot of different components. So it's either got to be lipid soluble, so small that it can get through, like water. Water's special, right? Don't bring water up. We'll get a little bit into that, but water is its own special thing. And then when we look at, if not, we got to have some other way to get it through there. So if you look at differences in concentration gradients, I like to use sodium. When we look at any given cell in its plasma membrane, there is generally more sodium outside the cell than inside. Extracellular sodium levels generally higher. Right? So there's a concentration difference. I need to slow down. I don't want to start being confusing. But if there's more sodium outside the cell membrane, plasma membrane, and there's less sodium inside, why doesn't the sodium just go down its concentration gradient? Concentration gradient means you're going to go from an area of higher concentration to lower concentration. Why doesn't sodium just equalize inside and outside of the cell?
Polarity.
Yeah, sodium has a charge, it's positive, and right, it's not lipid soluble. There's a barrier in the way. So the concentration gradient exists, but we need to open something like an ion channel to bring it in. See why I went back and said it that way? And so the concentration gradient will always be there. That's a chemical concentration gradient. Y'all threw in the fact that sodium is positive, right? So it has a electrical charge on it. And so you either have a basic concentration gradient, you have an electrical concentration gradient difference, positives versus negatives. Generally they exist at the same time because that's the electrochemical gradients. And we're going to really need these. Right? Hopefully I'm not getting too ahead of myself, but if I have a channel, an integral protein ion channel, like potassium pumps and things, sodium potassium pumps. If I have a channel that's in this plasma membrane, and it allows, if I open that channel, it allows sodium to go down its concentration gradient. Right? If there was no concentration gradient, I could open that channel all day long and nothing would happen. Like if sodium is already equal. I mean, I think you understand that, but I'm repeating that for a reason. Right? We still need to have a concentration gradient even if we open up an ion channel. You see what I'm saying? Nothing will move if there wasn't. Because we're going to use that for a lot of things. So many cells produce a lot of different gradients. Oxygen and sodium are more concentrated in the extracellular fluid. Well, we understand that. Oxygen's coming from the lungs, the It's traveling to these cells, it's in the extracellular fluid, so there's higher oxygen molecules out there, there's also higher sodium. Inside of the cell is going to be higher in carbon dioxide and potassium. Right. We're going to keep going through that. Also, the inside, we didn't discuss it a whole lot yet, but the inside of the plasma membrane is typically negatively charged. So if we were to draw that kind of plasma membrane that was here before, when we look at the inside, oops, really the way it looks is there are positive charges lined up extracellularly along the cell membrane, and then inside would be more negatively charged. I didn't mean to draw a straight line. So outside is generally more positive, inside is more negative. Right? That's part of the electrochemical gradient. And so anytime you talk about a concentration gradient, whether you're talking, a sodium molecule, whatever it might be, it's going to go downhill. It's going to go from an area of higher concentration gradient to lower concentration gradient. But if it's an electrical gradient, it's going to go from an area of positive versus negative charges. Right? So a positive charge, if you take two magnets and you take the positive side of one magnet and face it in this way, and you take the positive side of another magnet and face it in this way, and you bring them close together, what's going to happen? They don't go together. And it's going to push. Actually, that's a force you can measure, we'll see later, is there's a resistive force there. There's also a force, if I flip the magnet over, and now I have a negative side here and a positive side here, I get them close together, they're going to go together, right? That's how charges work. So depending upon how many positives you have outside versus inside, you also have a gradient. Most of the time, you don't just look at it as charges, though, individually, because it's going to be a charge associated with something like sodium. So down electrochemical gradients is generally what we see. All right. So, I'm talking a little bit outside of the slides today to give you more info up front. Is it seeming really confusing, or? It's just there's a lot we got to get to, and it fields questions now. But a lot of times when I talk to it that way in the beginning, I think when we get to the slides where we talk about that, that's when you go, oh yeah, now I see that's what we're talking about, and we can define it a little more. If you're getting confused so far, don't worry about it, because when we go through all of the different ways that this movement occurs, all of this should clear up. Right? Or if you need to today, go through that and read through it. Because tomorrow, I'm going to cut it early today, but tomorrow we're going to talk about what's the difference between a passive and active process and how do they occur, and we'll bring back all these little snippets I've been talking about today to help link that together, and it should make sense. If not, we'll stay on it in the morning until we get it. All right? If you start reading this stuff tonight, the biggest difference you need to pay attention to is passive processes do not use energy. Active processes must use ATP energy. That's where we're going. All right, any questions? All right, I don't think Rosario will be here till 10, but at times y'all's. All right, I'll give you over to your class so you can. I'm done for the day now. I know. People say often, mic drop, but